Well, hello everyone, and welcome to part 2 of chapter 4. I am your professor, Professor Bryn, and I'm looking forward to talking to you a little bit more about sensation and perception. I know that in week 4 you have many items on your to-do list and many concerns on your heart and mind. I just want to acknowledge and give you praise that you're doing a great job. I'm proud of you, and I'm so thankful to have you put the effort into this material. As you have an exam coming up, the time that you put in and the pacing that you put in and the growth mindset that you put in of learning these things, it will absolutely do you a benefit when we test for exam 1. Exam 1 will cover chapters 1, 2, 3, and 4. So what are we talking about today? Well, today in part 2, we want to talk about vision and hearing, as well as mention some of the other senses. There's so much about sensation that this lecture could be 10 hours long, no problem. And you're probably going, oh my gosh, please let it not be 10 hours long, right? Well, don't worry, it's not going to be 10 hours long. We're just going to go over the highlights, but especially focusing on vision and hearing. We'll also talk about depth perception and Gestalt psychology. And all in all, hopefully you're going to walk away with a greater appreciation for your senses and your ability to navigate the world around you. All right, so let's move forward and talk a little bit about the physical properties of light and by extension sound as well, but we'll start with light. So the physical properties of light and sound, so vision and audition or hearing, they both occur in the form of waves. So let's talk about wave forms, in particular as we understand that this is the physical property of the external world that is reaching our sense receptors. Two physical characteristics of a wave are amplitude and wavelength. Wavelength refers to the length of a wave from one of its peaks to the next peak. So amplitude is essentially the height of the wave, from its peak to its trough, so the entire height of the wave. Wavelength is directly related to the frequency of a given waveform, the number of waves that pass over a certain period of time, and it's often expressed in cycles per second. Longer wavelengths will have lower frequencies, and shorter wavelengths will have higher frequencies. So the visible spectrum of light is the portion of the larger electromagnetic spectrum that we can see. The electromagnetic spectrum encompasses all of the electromagnetic radiation that occurs in our environment, and that includes gamma rays, X-rays, ultraviolet light, the visible spectrum, the infrared spectrum, the microwave spectrum, and the radio waves. The visible spectrum in humans is associated with wavelengths between 380 to 740 nanometers. That's a really small distance because a nanometer is 1/billionth of a meter. We know that other species can detect other portions of the spectrum. For example, honeybees can see light in the ultraviolet range, and some snakes can even detect infrared radiation. So light that is visible to humans makes up a small portion of the electromagnetic spectrum. In humans, light wavelength is associated with the perception of color. Within the visible spectrum, our experience of red is associated with longer wavelengths. Greens are associated with more intermediate wavelengths, and blues and violets are shorter in wavelength. You can remember this Roy G. Biv, right? Red, orange, yellow, green, blue, indigo, violet. So that's the wavelength. Longer waves, meaning a lower frequency, are associated with the red part of the spectrum, and shorter waves associated with a higher frequency wavelength are associated with the blues and violets. So wavelength is perceived as hue or color. The amplitude of light waves is associated with our experience of brightness or intensity of color, with larger amplitudes appearing brighter. So since amplitude is measured as the height of the wave from the very top to the very bottom, amplitude is seen for us as brightness, perceived as brightness, with a higher amplitude indicating a higher brightness. Another property of wavelength has to do with purity, or the saturation of color. This has to do with the purity of the wavelength in such a way as a single waveform could be a very pure color, but multiple waveforms, multiple different waves at the same time, would be a less pure color. So let's imagine that you have red paint. Let's imagine that that red paint is a wavelength. It's just red, it's primary red, there's nothing added to it. If I add pink and black and a little bit of white, like I add different wavelengths into that paint, the paint has less purity than it had when it was just red. So, a mixture of wavelengths to us is usually perceived as the saturation of a color, with more consistent and similar wavelengths as being seen as more pure or more saturated, and a mixture of wavelengths as desaturated or a little bit less primary, a little bit less pure or saturated. Another way to think of that is that saturation has to do with the richness or the depth of a particular color. So an increase in purity or saturation means that the color has no white light in it. It's not mixed with other light types or variations. All right, let's talk about the visual system. Absolutely, the visual system constructs a mental representation of the world around us. It contributes to our ability to navigate the world and navigate through a physical space in which there might be multiple things to interact with or avoid or cling to, important individuals, objects, and situations. To begin, we want to talk a little bit about the anatomy of the visual system. The eye is the major sensory organ involved in vision. Light waves are transmitted across the cornea and enter the eye through the pupil. The cornea is the transparent covering over the eye. It serves as a barrier between the inner eye and the outside world, and it's involved in focusing light waves that enter the eye. The pupil is the small opening in the eye through which light passes, and the size of the pupil can change as a function of light levels in the surrounding environment. That size of the pupil can also change in accordance with emotional arousal levels within the person. When light levels are low, the pupil will become dilated or expanded. This allows more light to enter the eye. And contrastingly, when light levels are high, the pupil will constrict or become smaller to reduce the amount of light that enters the eye. The pupil's size is controlled by muscles that are connected to the iris, which is the colored portion of your eye. After passing through the pupil, light crosses the lens, a curved, transparent structure that serves to provide additional focus. The lens is attached to the muscles that can change its shape to aid in focusing light, and that light is reflected from near or far objects out in the world. In a normal-sighted individual, the lens will focus images perfectly on the small indentation in the back of the eye known as the fovea, which is part of the retina. The retina is the light-sensitive lining of the eye, the back of the eye. Now, the fovea contains densely packed specialized photoreceptor cells, and these photoreceptor cells are known as cones. They are light-detecting sensory receptors. The cones are specialized types of photoreceptors that work best in bright light conditions. Cones are very sensitive to acute detail, and they are able to provide spatial resolution. They're also directly involved in our ability to perceive color. While cones are concentrated in the fovea, where images tend to be focused, rods, another type of photoreceptor, are located throughout the remainder of the retina. And rods are specialized photoreceptor cells that work well in lower light conditions. And while they lack the spatial resolution and color ability of the cones, rods are involved in our vision in dimly lit environments, as well as in our perception of movement on the periphery of our visual field. Certainly, we've all experienced different sensitivities of rods and cones when making the transition from brightly lit environment to a dimly lit environment or vice versa. It's something that we talked about in part 1 of this lecture when we were talking about sensory adaptation. This has to do with the rods and cones adjusting their sensitivity depending on situations and depending on the conditions in which they're placed. Rods and cones are connected to retinal ganglion cells. Axons from the ganglion cells, they converge and exit through the back of the eye to form the optic nerve. The optic nerve carries visual information, so let's talk about that pathway. Light out in the world in the form of waves hits your eye. It gets focused by your pupil, it gets focused by the cornea and the lens. It hits an appropriate part at the back of your eye in the retina. The retina has rods and cones. Those are the photoreceptors or sensory receptors for vision. They're going to talk to other neurons essentially, and those neurons are going to communicate with the optic nerve. And the optic nerve is going to carry that visual information from the retina into the brain. Now, there is a spot in your visual field called the blind spot. Even when light from a small object is focused on the blind spot, we often don't see that object. And there's a really cool example in your book that I'd love for you to check out. It took me a little bit of time to actually get it to work. You have to be pretty close to the page to have it work, but take a look at that. I think you'll like it. We're not consciously aware of our blind spot for 2 reasons. First, each eye gets a slightly different view of the visual field. Therefore, the blind spots don't overlap. And secondly, our visual system will fill in the blind spot. This is like a top-down process, so that although we cannot respond to the visual information, we are not aware that the information is missing because of the top-down help from our brain. You should know that the blind spot is the place at which essentially the optic nerve is exiting the eye. So there's no rods or cones right there because they're just axons that are connecting the eye to the brain. So before we get a little bit farther in, let's talk a little bit more about rods and cones. Rods and cones get their name because they actually look like rods and cones. This picture from your textbook that I think is just so fascinating shows, you can see the cones sort of are pigmented a little bit pink in this picture, and the rods look red. What's important to remember is the retina is multi-layered. It's going to record light information for the purpose of converting it into neural information or action potentials to send to the brain. The retina and these rods and cones are going to allow us to take wavelength energy and convert it into electrochemical impulse energy, which is action potentials. And action potentials are the currency of the brain. As mentioned before, rods are sensitive to light. They're not great for color perception, but you have a lot of rods, nearly 120 million. They function and fire the best in low illumination or dimly lit environments. And they will absolutely change their sensitivity based on the environment they're in. As you remember, this is sensory adaptation. Cones are easy to remember that they're color vision because cones and color start with the same sounds. They operate best under bright light or high illumination. And you have fewer cones, but they are definitely best in daylight and in charge of color perception. In the center of the retina, you have an area called the fovea, as mentioned, which is only cones. And that is sort of the best, clearest, focused vision. It's just packed with cones and has really great acuity there. As mentioned, the blind spot is that area where the optic nerve leaves the eyeball. And this next slide walks us through the pathway or the visual pathway from the outside world of sensation to the inside world of visual perception. So let's walk through this process. Okay, so we have light out in the world, which is reflected off of objects and surfaces. It has a wavelength, it has an amplitude, it has a purity to it. It has a distance from us. So we're getting light information. That's the natural stimulus out in the world. It's going to go cornea, pupil, lens, and then retina. And that light, when it hits the back of the retina, is going to be engaging the rods and cones. And the rods and cones are the sensory receptors or the photoreceptors that are going to transduce that light stimulus into a neural impulse or an action potential. After the rods and cones, there's going to be bipolar cells. They're kind of like interneurons. They're going to connect with the ganglion cells. And the axons of the ganglion cells make up the optic nerve. And the optic nerve is what's going to take this information, which is now an electrical chemical impulse, an action potential, to the brain. The optic nerve from each eye merges just below the brain at a point called the optic chiasm. And the optic chiasm is an X-shaped structure that sits just below the cerebral cortex at the very front of the brain. At the point of the optic chiasm or crossover place, information from the right visual field, which comes from both eyes, is actually sent to the left side of the brain. And information from the left visual field from both eyes is sent to the right side of the brain. So this slide essentially walks us through and shows us how information comes in to your left or right visual field of both eye. And because of the optic chiasm, the information crosses over to the other hemisphere. Once inside the brain, the visual information is sent via a number of structures to the occipital lobe at the back of the brain. So you know the occipital lobe in the very back is responsible for vision. Visual information might be processed in several different pathways at the same time. One pathway might be identifying objects, or another pathway might be identifying distance or location of objects. So, for example, it's important for your brain to both identify what's coming towards you and how fast it's coming towards you and from what direction it's coming in space. So to review, after information has come into your eye, gone to the retina, rods and cones, bipolar cells, ganglion cells, optic nerve, it's going to essentially cross over at the optic chiasm and go essentially either to the right occipital lobe or left occipital lobe. The information will come in via the optic nerve, and at the optic chiasm, it's essentially going to cross over. So if information came into your left visual field of both eyes, it's going to go to the right hemisphere. If the information came into the right visual field of your eyes, it'll cross over at the optic chiasm to the left hemisphere. Before getting to the occipital lobe, either right or left occipital lobe, the thalamus is the sensory relay system, so it's going to take in and process some of that information before sending it on to the occipital lobe. And in the occipital lobe, you have visual cortex. Again, like I mentioned, there's identification parts of the visual cortex of knowing what is coming at you, and also location or depth perception or distance information in that cortex as well. So, retina, optic chiasm, thalamus, occipital lobe, primary visual cortex. That visual cortex has feature detectors. Even that area of your brain has specialized cells for responding to specific features of a stimulus, like edges and movement, or even things like size, shape, color, lines, or angles. And the brain in the visual cortex undergoes something called parallel processing and binding. That allows us to know what's coming at us and from where it's coming. The what and the where pathways, essentially. Why would we want to have parallel processes? Well, we'd want to have quick integration of visual stimuli. That's good for our survival. It's important to know what and where as quick as you can so you can respond as quick as you can. So I want to move forward and talk a little bit about color vision. In order to talk about color vision, we have to talk about theories of color vision. It really is quite remarkable to think about the fact that the two theories that I'm going to explain to you were proposed far before there was the science to be able to know these biological micro places or these sense receptors. The people that theorized this absolutely had tremendous insight before their time. So let's talk about two different theories. This is going to be the trichromatic theory first, and then the opponent process theory next. According to trichromatic theory, and this is known as the Young-Helmholtz theory of color vision, there are 3 receptors in the retina that are responsible for the perception of color. One receptor essentially is sensitive to green, another to blue, and a third to red. And the combination of these 3 colors produces all of the colors that we're capable of perceiving. Researchers suggest that people are able to distinguish between as many as 7 million different colors. It's amazing to think that in the 1800s, Young is proposing in his research that the eye contains different photoreceptor cells that are sensitive to different wavelengths of light in the visible spectrum. Later, in the mid-1800s, Helmholtz expands on Young's theory and suggests that the cone receptors of the eye are either short wavelength for blue, medium wavelength for green, or long wavelength for red, and they're most responsive within those short, medium, or long wavelengths. So it's not really that the 3 cones respond to 3 colors, they're responding to 3 different wavelengths of light. Short for blue, medium for green, and long for red. Helmholtz proposes that it's the strength of the signals detected by those receptor cells that determined how the brain interpreted color in the environment. Their combined work essentially becomes the Young-Helmholtz theory of color vision, or as we're going to refer to it, the trichromatic theory. The perception of color by the brain requires input from at least 2 different types of cones. So the brain must interpret information about both the wavelength and the intensity of the incoming stimulation. By comparing input from each cone that's been stimulated, the brain can interpret color and the color of the source of that stimulation. So color blindness, therefore, has to do with some sort of deficiency in one or more of the cones in the type or system of cones. In the past, the trichromatic theory was often presented as competing with this next theory that I'm going to talk to you about, which is called opponent process theory. But today it's believed that both of the theories, trichromatic and opponent process, can be used to explain how color vision operates, and that each theory applies to a different level of this visual process. So the trichromatic theory explains how color vision operates at the receptor level, the level of the rods and cones. And the opponent process, which we're going to explain in the next slide, it offers an explanation on how color vision operates at the neural level, or the brain level. Okay, so we know that the trichromatic theory explains one part of this process, and that's the process focusing on the photoreceptors, the rods and cones, in the back of the eye on the retina that send signals to the brain. I want to talk now about the opponent process of color vision. Okay, in order to make sense of this theory, we have to do a little activity. And in this activity, I'm going to show you a slide, and it is a square in which there is a black dot in the center. You're not going to focus or look at anything else in the picture. I just need you to focus on the black dot in the center of the picture, okay? So we're going to stare at this black dot. Your job is to not blink, to not move your eye, to not move your head. You just really need to stare at the black dot for at least 25 seconds, okay? So I will switch to this slide. Get yourself comfortable. Put yourself in position. You're going to stare at the black dot, 30 seconds. Try not to blink. Ready, set, go. Well, what do you think? Did you see the after image? There is a unique thing that happens in color vision, and it leads us to our next theory, which is the opponent process theory. one of the things that had been noted in regard to the development of opponent process theory is that there was something called after images. And I don't know if it worked for you. You can go back and try again. It should work. But an after image describes the continuation of a visual sensation after the removal of a stimulus. So if you stared at that black dot and then looked away from it, you should have seen sort of the opposite colors in that color wheel. When color is involved in the stimulus, like it was for you guys, the color pairings identified in the opponent process lead to a negative after image. So Herring, the researcher that has to do with opponent processes, he notices this negative after image effect. And according to this theory, the opponent process theory, color is coded in opponent pairs. Your book talks about them as essentially being 4 different systems. So let me talk about how your book talks about it, and then I'm also going to talk about how we talk about it sort of now in the research. So your book talks about 4 cones in sort of opposite pairs. There's a blue-yellow or a yellow-blue and a red-green or green-red. And those are essentially opponent pair cones. So those cones that are in opposite pairs, blue and yellow, red and green, those pairs are wired in the visual cortex in such a way as only one of the colors can fire at the same time. So you stimulate the blue frequency of the blue cone, a yellow cone that's paired with it is less active, has a lower frequency. It's like a metaphorical seesaw. One goes up, the other goes down. One goes down, the other goes up. This opponent process can explain negative after images because that swing of back and forth speaks about the opposite cone after it's been suppressed. When it's free to fire, it's able to fire, which is why you see a negative after image. Currently understood, the opponent process theory of color vision suggests that our ability to perceive color is actually controlled by 3 receptor complexes with opposing actions. These 3 receptor complexes currently understood would be red-green, blue-yellow, and black and white. According to the opponent process theory, these cells can only detect the presence of one color at a time because the 2 colors oppose one another. You don't see greenish red because the opponent cells can only detect one of those colors at a time. So the opponent color process works through a process of excitatory and inhibitory responses. This is our metaphor of the seesaw, with the 2 components of each mechanism opposing one another. For example, red creates a positive or excitatory response, while green creates a negative or inhibitory response. One goes up, the other goes down. These responses are controlled by opponent neurons, which are neurons that have an excitatory response to some wavelengths and an inhibitory response to the wavelengths in the opposite part of the spectrum. So how does this system explain after images like the ones that we've tried in this lecture? Well, essentially what happens is you're fatiguing one of your cone pairs. And when you shift your perspective to the opposite image or a blank image, the other cell in the opponent pair is now able to go up the seesaw. So if you've been staring at a red dot and the red has been firing, the very second we go to just a gray background, the green now in the seesaw, it has not been active, but green now gets to pop up in the seesaw, and so you see green. When you shift your focus, the cells that have been firing are no longer able to fire, so you see the opponent color. As a result, you see the after image of the opponent pair. So to review, according to the opponent process theory, color is coded in opponent paired neurons, black-white, yellow-blue, and green-red. The basic idea is that some of the cells of the visual system are excited by one of the colors and wavelengths and inhibited by the other wavelength, and they can't both fire at the same time. So a cell that was excited by wavelengths associated with green would be inhibited by wavelengths associated with red, and vice versa. These two theories, overall, trichromatic and opponent process, they're not mutually exclusive. They just apply to different levels of analysis or different levels of the nervous system. For visual processing on the retina, trichromatic theory applies because the cones are responsive to 3 different wavelengths that represent short, medium, and long wavelengths. But once the signal moves past the retina on its way to the brain, the cells respond in a way consistent with opponent process theory. Both theories are needed to account for the complexity of color vision. To review, the trichromatic theory explains how the 3 types of cones detect different light wavelengths at the level of the retina, while the opponent process theory explains how the cones connect to the ganglion cells. You know, the ganglion cells are going to connect to the optic nerve. These ganglion cells are where the opposing elements inhibit each other to determine how color is perceived. Okay, so we're still talking about the visual system and how important the visual system is. Now we want to talk about Gestalt psychology. Gestalt is a German word that means configuration, and as a perspective, it has to do with understanding whole events and whole items. So your brain doesn't just add a whole bunch of small things together to create a big object. The big object often is more meaningful than its component small little parts. So my understanding of a visual stimuli as a cat is more than just me having feature detectors computing legs plus whiskers plus fur plus tail. That the cat is not just a bottom-up process, but a top-down process as well, a meaningful top-down process. There are some Gestalt principles that I want to show you. We're going to take a look at figure ground, closure, proximity, and similarity. All right, so let's start with closure. So on this slide here, you can see that closure and the principle of closure is that if I ask you what the orange item is or the orange shape is on the top left, you're going to tell me triangle because your brain can see that that's meant to be a triangle. You're not confused by that. And the other shapes, despite not being filled in, your brain is able to close or add closure to make sense of them. Your brain wants to see the whole and is able to see the whole. So that is a top-down or Gestalt process. Let's go over to proximity. So the Gestalt principle of proximity is that things that are clustered together are often seen as being connected with one another. So if I ask you how many groups are in the proximity example, most people are going to say 4 groups of 4. They're going to say that they cluster together there. Proximity allows your brain to put things together that are close together in proximity, seeing them as importantly connected. The Gestalt principle on the bottom of similarity shows us that our brain really likes to see either columns or rows of items. You're either on the left-hand side seeing columns of circles and squares, or on the right side of this picture, you see rows of circles or squares. Your brain puts the similar items together. It makes sense of the world better that way. Closure, proximity, and similarity are Gestalt principles that allow us to make quick decisions in the world about a world that has many top-down configurations and important whole gestalts. All right, so let's talk about figure-ground relationships. So what is this a picture of if the background, if the wall is white? If you're looking at this picture and you're imagining that there's a white wall in the back, what do you see? Or if I was to tell you that the wall behind this item is purple, what do you see? So whether or not the background is white or the background is purple, will change how your brain sees the items, whether it's two faces or it's a vase. Figure-ground relationship. All right, let's move forward and talk a little bit about depth perception. So our ability to perceive spatial relationships in 3D is known as depth perception. With depth perception, we can describe things as being in front, behind, above, or to the side of other things. Our world is three-dimensional, so it makes sense that our mental representation of the world also has three-dimensional properties. We use a variety of cues in a visual scene to establish our sense of depth. Some of these cues are binocular cues, which means that they use both eyes. And some of these cues are monocular cues, meaning that they only involve one eye at a time. So binocular vision really relies on something called binocular disparity. This means that despite you having two eyes, each eye has a unique representation because they're slightly differently spaced, right? The left and right eye don't have a perfectly similar image that they're taking in. The disparity between the two eyes and the combination of the information streams from both retinas, it's used for depth perception purposes. So the disparity or the convergence provide information about how far away an item or object might be. And absolutely, we rely on binocular cues to experience depth. But we can also perceive depth in a two-dimensional sense as well. When we do this, we're relying on monocular cues, or cues that only require one eye. So let's talk a little bit about some monocular cues for depth. Okay, in this picture we have two monocular cues at play: familiar size and overlap. In regard to familiar size, this has to do with learned experience. So just in the fact that I have top-down information about the fact that little girls are taller than paintbrushes, the familiar size of these items leads me to know that these paintbrushes are in front of the little girls. Obviously, in the picture, they're much larger than the little girls, but my brain is not confused. My brain knows that the paintbrushes, which are smaller, are closer to me than are these two little girls. Another monocular cue that I have is overlap, in such a way as I see that the paintbrushes are overlapping the girl that's in the middle. Those are two monocular cues that I can derive depth perception from. I don't need both eyes to see that the paintbrushes are closer to me than are the little girls. Another monocular depth cue could have to do with shading. I'm able to see, just from variations in color and shade, the depth and placement of certain items. So this is the 2D picture, but I'm able to sort of see where the armpit is and where the face is and where the chin is and where the nose is, and you would get to the nose before you would get to the ear. It shows me which orientation this person is facing, which eye is closer to me and which eye is facing away from me, which gives me information about orientation. And despite this being a 2D picture, I'm able to see monocular cues for depth. A couple other monocular cues, and we can see in this picture here: height in field and linear perspective. Linear perspective refers to the fact that we perceive depth when we see two parallel lines that seem to converge in an image. And you can see this essentially from the walkway that is converging towards the back of the picture. That would be linear perspective. Height in field also gives us some monocular cue for depth, such that I'm able to see that this lady in the front, she looks like she's closer to me than the people that are in the back picture. There's a guy with kind of a red outfit in the very middle back. And I'm able to see, because of the height in field, that she is closer to me than the person that's in the very middle of the picture in the red. Two monocular cues in this picture: height in field and linear perspective. Here's another monocular cue for depth, and that has to do with texture gradients. So if I was to ask you which square here is the closest to you, you'd probably pick this square on the bottom left, right? It's the biggest, and in this texture gradient, it looks like, despite it being a 2D picture, it looks like this pattern is tilting and sort of farther away in the back right corner because of the texture gradient, another monocular cue for depth. Your book also talks about motion perception. Well, humans have very specialized motion detectors. Those motion detectors are not in the retina, they're actually in the brain. They're specialized neurons that give feedback from our body and also receive lots of cues in the environment. So what's neat about them is that they're very, very able to make quick assessments of motion. It's important for you to be able to see motion quickly. If something's coming at you, it's important for your brain to respond actually pretty quick. And that's why you have motion detectors. But they're pretty sensitive, which means that sometimes your motion detectors go off and fire. They fire an action potential, even when there's not motion. And that is an example of apparent motion. So this picture that you're looking at, if it seems a little bit like it's moving, that's your motion detector system just being a little bit sensitive to the stimuli around you, because that system is set to be somewhat sensitive. And this particular visual illusion for motion is working because of these specialized motion detectors that you have in your brain. Before we move on to hearing, I definitely want to direct your attention towards a concept called perceptual constancy. And this is around page 124 of your textbook. There's a really cool quote, and I wrote it down, that I want to bring to your attention. It says in the book on page 124, experiences are made sensible through perception. So top-down processing allows us to know and interpret the things that are in our world. We're not constantly surprised by a shifting visual world because of perceptual constancy. And that's the recognition that objects are constant and unchanging, even though the sensory input from the retinal firing about those objects is constantly changing with the reflected light, with where you're standing and how you're moving and what's going on. If your best friend that you're talking to outside steps in the shade, sort of steps to her left and she goes under the shade to get out of the sun, you don't go, oh my gosh, who are you? Hi, my name is Bryn. Like, you don't reintroduce yourself to her because you know that despite the coloring changing, she's still your friend. So perceptual constancy allows us to stay constant and constantly understanding a visual world around us that is constantly changing. Experiences are made sensible through perception. More specifically, top-down processing. All right, let's move forward towards properties of sound and the sense of hearing or audition. And since we're dealing with wavelengths, we have similar properties to light that we saw earlier in this lecture. So let's go through wavelength, amplitude, and timbre or purity. So sound waves are vibrations in the air that are processed by the auditory system. Wavelength, again, is the distance between two peaks in a waveform. And wavelength, for our purpose, determines the frequency or pitch of a sound. A higher frequency wavelength equals a higher pitch of sound. And there are some wavelengths that you can't even perceive. They're outside of your perception. Dogs can hear things and cats can hear things that we can't hear. They are responsive to those wavelength and frequencies that we're not. Amplitude has to do with the height of the wave, so the very peak of one wave and the trough or the bottom of the other wave, how high that wave is. And amplitude is perceived as volume or loudness. We use decibels, right? dBs. So the higher the amplitude, the higher the volume, which is perceived as a louder sound stimuli. And like talking about purity or saturation in vision, sound also has this mixture of wavelength dynamic as well. So we use a term for purity or complexity of the waveform. In sound, it's called timbre. This is the saturation or the clarity of a sound. So something is very clear if it has a very simple waveform. And something is less clear if it has more complexity, where it has multiple frequencies and waveforms attached to it or going on at the same time. So something with a clarity of sound might be a very skilled singer singing very clear and very high by herself. Whereas if you add 3 or 4 people with her and maybe they're not as expert in singing, if they all sing at the same time, some of the clarity or purity or saturation is going to diminish. The timbre or clarity of the sound will be reduced. Okay, so let's move forward and review the anatomy of the auditory system. And I direct you to the picture here on this slide as we just walk through the story of sound, going from the outside world, being sensed by your sense receptors, and being interpreted and perceived by the brain. The ear can be separated into multiple sections. The outer ear includes the pinna, and that's the visible part of your ear, the auditory canal, and the tympanic membrane or eardrum. The middle ear contains these 3 tiny bones, and that is the hammer, anvil, or stirrup. The Latin names of those things are malleus, incus, and stapes. The inner ear contains the semicircular canals, which are involved in balance and movement, the vestibular senses, and the cochlea. The cochlea is a fluid-filled structure. It's a rolled structure. It looks like a snail that contains the sensory receptor cells. These are hair cells of the auditory system. Sound waves travel along the auditory canal and strike the tympanic membrane, causing it to vibrate. This vibration results in the movement of those 3 small little bones. As the hammer, anvil, and stirrup move, that stirrup presses into a thin membrane of the cochlea known as the oval window. As that last bone, the stirrup, presses into the oval window, the fluid inside the cochlea begins to move, which in turn stimulates the hair cells, which are the auditory receptor cells of the inner ear embedded in the cochlear membrane that we call the basilar membrane. The basilar membrane is a thin strip of tissue within the cochlea. So the activation of these hair cells on the basilar membrane is a mechanical process. The stimulation of the hair cell ultimately leads to the activation or neural firing of that cell. As hair cells become activated, they generate these neural impulses that travel along the auditory nerve to the brain. So auditory information is shuttled from the cochlea into the thalamus and finally to the auditory cortex in the temporal lobe of the brain for processing. And like the visual system, there's evidence that suggests that the information about auditory recognition and localization is processed in parallel streams of what is this and where is this. So what's the pathway of auditory information? Sound waves out there in the real world, being funneled through the pinna and the eardrum and those 3 small bones of the middle ear, through the oval window into the fluid-filled cochlea. That cochlea is lined with a basilar membrane, and the basilar membrane, which is the inner wall of the cochlea, has the sensory receptors for hearing, the hair cells. When the hair cells vibrate and basically fire their action potential, they're going to be converting those vibrations and those waveforms of sound pressure from sound pressure into action potentials. That neural impulse gets sent to the brain by way of the auditory nerve. It'll go towards the thalamus for processing and then to the temporal lobe, which deals with hearing and being able to localize and identify what and where the sound is. That's a parallel process of information. We're able to find out where it is and what it is rather quickly. Before moving on, I want to talk a little bit more about localizing sound. The ability for us to locate sound in our environments is a really important part of hearing. Like monocular and binocular cues about vision and depth, the auditory system also uses monaural or one-eared or binaural two-eared cues to locate sound. Each pinna interacts with incoming sound waves differently, depending on the sound source relative to your body. This interaction provides a monaural cue that is helpful in locating sounds that occur above or below or in front or behind us. So you can see that even one ear can be able to orient itself in space and figure out where a sound is coming from. Binaural cues also provide information about the location of a sound by relying on differences in the vibration patterns between the two ears. So there's two types of binaural cues. There's intensity differences between the two ears. So if the song is coming from my right, then my right ear is going to have the louder amplitude of the sound, right? And the left will have a slightly less intense form or volume of the sound. And also timing differences between the arrival time of those frequencies at each ear. If a sound in a song is coming from my right side, it's going to hit the right side before the left ear. So that level or intensity difference refers to the fact that a sound coming from the right side of your body is more intense at your right ear than at your left ear because of the attenuation of the sound wave as it passes through your head. And the timing difference between the two ears refers to the small difference in the time at which a given sound wave arrives at the right or left ear. So localizing sound involves the use of both monaural, one-eared, and binaural, two-eared cues. Moving forward, just like with color vision, there are two theories of hearing that help us explain how we're able to perceive pitch: place theory and frequency theory. Different frequencies of sound are associated with differences in our perception of the pitch of those sounds. Low frequency sounds are lower pitched, and higher frequency sounds are higher pitched. So how does our auditory system differentiate among these various pitches? Let's start with place theory. The place theory of pitch perception suggests that different portions of the basilar membrane are sensitive to sounds of different frequencies. More specifically, the base of the basilar membrane, that's the place that's right by the stirrup, right by those little bones of the middle ear, responds the best to high frequencies. And the tip of the basilar membrane, that would be if you were to unroll the cochlea, it would be the far end, the unrolled end. It responds best to low frequencies. Therefore, hair cells that are in the base portion of the cochlea are labeled as high pitch receptors, and those in the tip of the basilar membrane would be labeled low pitch receptors. Now, place theory best explains high frequency sounds, and it doesn't explain the whole range of what we're able to hear. Therefore, it's not a complete theory for the entirety of our pitch perception ability. So we have another theory called the frequency theory of pitch perception, and this has two different parts. So in one part of this theory, what is proposed is, okay, so you've got place theory, that's great, but maybe we perceive pitch because of frequency of how fast the nerves are firing. Nerves that fire faster would be a higher frequency. The problem with that, of course, is that because nerves and neurons are all or none, they either fire or they don't, and they have a limitation of how many times they can fire before they have to rest. So just the frequency of one neuron can't explain all the frequencies that you can hear. Therefore, there is this volley principle in such a way as whereas one neuron might not be able to fire faster than 1,000 times a second, if you have 10 neurons and they're all firing at that level, that can get me to a much higher frequency than just one cell by itself. In reality, both theories explain different aspects of pitch perception. Together, place theory and frequency theory allow us to cover for and account for high frequency and lower frequency sounds. All right, we're doing awesome. You're doing so great. So let's talk a little bit about taste and smell. These are essentially our chemical senses. Taste and smell are called chemical senses because both of those senses have sensory receptors that respond to molecules in the food that we eat or the air that we breathe. And there's absolutely a pronounced interaction between these two chemical senses. So let's talk a little bit about taste. So you might have learned in elementary school that there are 4 types of taste: sweet, salty, sour, and bitter. Well, there's evidence that there's a 5th taste called umami, and that's associated with a taste for monosodium glutamate, stuff like soy sauce and savory things like cheese, parmesan, soy, fish. So in taste, molecules from the food and beverages that we eat dissolve in our saliva and interact with the taste receptors on our tongue, in our mouth, and throat. Taste buds are formed by groupings of taste receptor cells that have hair-like extensions, and those extensions protrude into the central pore of your taste bud. Those taste buds have a life cycle of about 10 days to 14 days. So even destroying some by burning your tongue won't really have any long-term effects, they just grow back. Although it does sound like they become less sensitive as you age. Taste buds are composed of a number of individual taste receptor cells that transmit information to our nerves. So taste molecules will bind to those receptors and cause chemical changes within the sensory cell that result in neural impulses being transmitted to the brain via different nerves, depending on where the receptor is located. Taste information goes to the medulla, thalamus, limbic system, and even the gustatory cortex, which is underneath the frontal and temporal lobes. Now, smell, or olfaction, involves olfactory receptor cells, and those cells are located in the mucous membrane at the top of your nose. Very small hair-like extensions from these receptor cells serve as the sites for the chemical molecules dissolved in the air to interact with the chemical receptors located on your nasal passage cell receptors. Once an odor molecule has bound to a receptor, chemical changes within the sensory receptor result in signals being sent to the olfactory bulb. And that bulb-like structure is at the tip of your frontal lobe, where the olfactory nerves begin. From the olfactory bulb, information is sent to regions of the limbic system and then to the primary olfactory cortex, which is located very closely to the gustatory cortex that we talked about in regard to taste. That is underneath the frontal and temporal lobes. Now, all of your other senses go through the thalamus except for smell. So we call smell special. It has a direct line to the brain, a direct perception. So you have olfactory epithelium to the receptor cells, to the olfactory nerve, to the temporal lobe and limbic system, straight to the brain. All right, let's move forward and talk a little bit about touch, temperature, and pain. So a number of receptors are distributed throughout your skin, and your skin is the largest organ of your body. And those receptors are responsive to different types of touch-related stimuli. And that touch-related stimuli might be temperature perception or pain perception or pressure. Sensory information collected from these receptors travels up the spinal cord and is transmitted to regions of the medulla, thalamus, and ultimately to the somatosensory cortex, which is located in the parietal lobe. As you remember, your somatosensory cortex is like a map of your body. So it has an area for your right arm. It has an area for your left toe. It has an area of your hip to the left side. So it pretty quickly is able to isolate and tell you where the issue is, where in your body something is being touched, getting warmer, or in pain. Your book also talks about temperature and pain receptors, and I think this is really cool. You have specific receptors, sensory receptors for temperature. They're called thermoreceptors, and they are warm and cold receptors. But sometimes they have a paradoxical reaction, and they can sometimes, when they're firing both at the same time, sometimes they can be perceived as hot, that's paradoxical hot, and sometimes when they're activated both at the same times, you can be perceived as cold, which is called paradoxical cold. So do yourself a favor and take a look into page 133. There's some good discussions, a little bit more about the skin senses, including nociception, which is pain perception, and you can learn a little bit more of the story there. The final thing that I want to talk about has to do with the vestibular sense, proprioception, and kinesthesia. So the vestibular sense contributes to our ability to maintain balance and body posture. So what's your posture like right now? Are you laying down? Are you sitting in a chair? Are you standing? Are you moving? The vestibular organs are fluid-filled and have hair cells very similar to the ones found in the cochlea of the auditory system. The vestibular organs and those hair cells in there, they respond to movement of the head and to gravitational forces. When these hair cells are stimulated, they send signals to the brain via the vestibular nerve. So the major sensory organs of that vestibular system are located next to the cochlea, and they're in the inner ear. In addition to maintaining balance, the vestibular system also collects information that's critical for controlling our movement and the reflexes that move different parts of our bodies to compensate for changes in body position. Therefore, both proprioception, which is perception of your body's position, and kinesthesia, the perception of your body's movement through space, interact with the information provided by the vestibular system. This helps you understand where you are in the world, how you're moving, and if you're balanced. These sensory systems also gather information from receptors that respond to stretch and tension in the muscles, joints, skin, and tendons. That proprioceptive or kinesthetic information travels to the brain via the spinal cord. And several different areas in your brain, including the cerebellum, receive information from and send information to the sensory organs of those proprioceptive and kinesthetic systems. This is how you can move and accelerate and dance and jump and lay down and drive in the real world without it being total chaos or you falling over every 5 seconds. All right, you guys. Well, you did a great job today. I'm super proud of you. I know that it's a lot. I know you have a lot of information in front of you, but I just want to affirm for you that I'm proud of you. You just keep going one little chapter at a time, one little task at a time. You're doing great. And as always, if you have any questions or want to talk to me or want to schedule an appointment to go over some stuff, let's talk about it. I love this stuff. I can't wait to talk to you about it. As always, it's a pleasure being your professor. So wherever this lecture finds you, remind yourself that you're awesome. Just get to the next thing. You're going to be okay. All right, you guys. That's all I have for you. I'm so proud of you. Have a really good week. I'll catch you next time.